Thank you, Alex. Thank you for this opportunity to be here again. How many people are ready to go home? Just raise your hand. No one? I'm the last talk, so I'll try to make this quick. Most of you have heard what I have to present. It's a pleasure to be here. Thank you, Sergey. Thank you, Alex. I've spoken about this before. I think we're changing our mentality like the artists did 500 years ago. As you know, in this famous figure, a child was depicted just like an adult. And it was about 500 years ago when we started drawing children like they are. I think in neurooncology and in research, we still are thinking of most of the pediatric diseases as adult. We need to segregate those, and you've heard most of the talks today, the ones given by Dr. Packer and others, that pediatric diseases, specifically pediatric cranial tumors, are different than adults. We have to start thinking differently about them. Why is it so complicated? You've heard about many of the diseases today, low-grade gliomas, medulloblastomas, and our difficulties with treating them. I think the complexity comes from the biology. We all know we have 20 to 25,000 genes, but at the RNA level, these genes transcribe about 100,000 transcripts. These are alternative splicing of the RNA. And the RNAs are actually translated into protein, which is post-translationally modified in making over 1 million molecules. And again, with most of the diseases that were discussed today, is a signaling molecules. It's typically a phosphorylated nuclear membrane protein that is dysfunctioning and is causing the tumor. It's really difficult for us to understand the function of over 1 million molecules, despite all of the research that Alex is doing here and your lab colleagues, Mila and other colleagues are doing. Again, if you look at the genome, these two different species, this is one of my favorite figures. I think some of you have seen it here before. If Alex takes this caterpillar to the laboratory and do genome analysis, he would never be able to understand the biology of the butterfly. They have exactly the same genome, but they have a different proteome. And again, I think in most of the talks that you've heard today, we constantly look at genome, which is a good thing, but we are not looking at the proteome, and I'm not going to address that today. My point is that we are not truly studying biology of the cancers because we don't have all of the tools. Again, a center may be good in genomics, but not in proteomics, but maybe also not in signaling molecules. Again, radiographic. Our colleagues show radiographic images of the brain tumor. It's a black and white. White is tumor, black is empty space. This is how we typically look at the tumors. We're not delving into the biology of the tumors, and we want to do it. At Children's National, I run the scientific part of the Brain Tumor Institute, and we have areas of research, medulloblastomas, low-grade, NF, and DIPG. My specific interest is DIPG, although I have a lab that is active in all of them. We have developed a Biorepository. We work very close with the neurosurgeons at the Children's National, such that every time they have a surgery, if there's anything that we can bank, we bank that sample. Even when a surgeon uses a catheter to take a biopsy, and that biopsy is not enough for us, we typically take that catheter and we wash that off. We typically get cells from that catheter, and we can store those. So we've gone from really having 8 samples for DIPGs to over 1,000, this has not been updated, to over 1,200 patients banked. This is over 5,000 samples that we have banked over the past 10 years. And it's been really working closely with the pathology and also with the neurosurgery. Our samples include astrocytomas, ependymomas, neuronal, embryonal tumors, and craniopharyngiomas. Again, if there is a specific laboratory or scientist or researcher who has a particular interest, the bank is in the service of that particular investigator. We do collect CSF, frozen blood, FFPE, and urine from all of our patients who have a brain tumor who come in for treatment. We've also started collecting cheek swabs from the parents, because some of these tumors may be driven by mitochondrial DNA, which we don't know. Again, we don't want to wait another 20 years and say, we wish we had collected samples from the parents. We're collecting from the siblings and also from the parents. Again, my emphasis today is on DIPGs. I guess the audience here knows about DIPGs. It's really one of the most terrible type of brain tumors. They give 15%. Dr. Packard today showed a slide that had 10% survival. They really don't have 10% survival. I've known of one DIPG patient, one, who has survived beyond 5 years, and I know she does not have histone mutation by liquid biopsy, which I will explain today. And again, your colleagues Mila and Alex are developing that here at Rogachev Institute. There is an idiom search in Google. Have you guys, if you haven't tried it, is a trend in Google. You can put any word and look at that word throughout the history. If you search for the word DIPG in Google Trend, you will see there's actually nothing mentioned about DIPG before 2006. But after 2011, there's actually a spike in the number of times that the name DIPG has come up. And Google is actually looking at all of the published information. All of the Oxford laboratory has been scanned by Google to looking at everything. So this is when the word DIPG started coming up in the searches. If you look at this, why was it? Why did it really, the word Google DIPG started coming up? The first models, the first rat models, they started developing in late 2000. And then we had cerebellar xenografts that were developed. And then we had DIPG xenografts. As soon as we have DIPG xenografts, there are more numbers of hits on the word DIPG that start showing up in the history. And you have DIPG cell lines that were developed. It was a huge breakthrough. This is not an ancient history. We're really talking about the past 6 to 7 years. DIPG cell lines followed by a number of scientific publications. The red line here is showing the number of publications. This is scientific publications. Before that, we have hundreds of clinical publications showing that clinical trials do not work in DIPGs, indicating that all of the trials have failed. This is scientific papers discussing the molecular basis of DIPG. So it really shows that you need to have a good laboratory in order to do a good clinical work. The same thing that a good restaurant has a very good kitchen. A good hospital needs to have a very good research. And you can divide this into early animal phase models, robust preclinical models, and I think we've entered an era which is clinical translation. We have many models for DIPGs, for high-grade gliomas. We have many cell lines that need to be translated, and I will talk about some of the translations that we are doing. We've identified some of the mutations associated with DIPGs. These are histone mutations, ACVR, P53, ATRX, and so on, PPM1D. And you can actually divide the brain into different zip codes where the mutations could happen. Within the brainstem, you have ACVR and histone 3. The histone 3 mutations, if it is a G34, is almost always a cortical tumor or a midline. It's not, or a thalamic. It's almost never a brainstem. And WHO has changed their grading specifically based on histone. You have a series of mutations, TP53, ATRX, and MYC-N, which could happen in the cortex or they could be shared with the brainstem. But histone tends and has been shown to be driving specifically within the brainstem. Again, the clinical translation phase, and this is what comes here to the clinicians and who should take advantage of what has been done in the lab to take it to the clinic. We need to do rapid expansion of preclinical models, which again, Dr. will be working on here, fully characterize this and also integrate this across species and models. And share data and resources. I think that's one of the reasons that we are here, to share data and resources. Because again, a child who has a tumor, it doesn't matter where they are in the world they have. The first thing that we've done with our samples was to understand the geneity. One of the questions that neurosurgeons have is that if they can get into the tumor with convention-enhanced delivery, would the tumor, if you look at the right side of the tumor to the left side of the tumor, is it the same or not? Does it have the same composition of mutation across the tumor? What is the spatial distribution and fidelity of the IPG? Do biopsies really represent tumor biology? We are taking biopsies. Are they really representing them? And what are targetable mutations? So we've collected a host of post-mortem samples from patients, We have one of the most active laboratories that collects post-mortem from DIPG patients. They actually are brought to our hospital and we take the brain to study. We collected samples from all over the brain. This is a sagittal section of the patient post-mortem, and we collected samples from everywhere within the brain. Surprisingly, you can actually detect tumor cells all over the cortex at the post-mortem level. If you do histological staining, you can actually see it. By the time of death, the tumor has spread beyond the brainstem. So one of the first things that we found, this is an evolutionary of the tumor. If you look at the brainstem, the early events, again, has been calculated bioinformatically. You have histone 3 and ACVR for this particular patient. As the tumor grows to the thalamus, there is a new mutation, PIK3CA, that is coming up. There's another mutation that is also arising in the medulla as the tumor is growing down, which is MAX. But altogether, what we have shown is that the major mutations, the major driver mutations for these patients are all the same. But one of the biggest criticisms that we always get for that work is that it's all post-mortem. Is post-death. So what would happen if you would actually take a biopsy from a patient with DIPG up front before they have received any mutation, any treatment? And we have done this through Pacific Pediatric Neyroncology Consortium, our hospital and the hospital at UCSF. We take biopsies of DIPG patients up front. Typically 5 to 8 biopsy cores are taken for molecular studies, but also for making preclinical models. This is the sketch of how the trial is done. If the patient is eligible, the biopsy is taken. There's an exome expression profiling, whole genome, and RNA profiling. There's a report. We have a specialized tumor board that sits and looks at the mutations, and the patient will be given a drug basket specifically for those drugs. In the research lab, we make xenografts, and also we take circulating tumor cells, which I will explain. So it's a research, but also a clinical trial specifically for DIPG where upfront biopsies are taken and the drug is given specifically based on the mutation. This is the first result. This paper will be published within the next few months. These are the mutation profile of our patients up front. Again, if the patient has not seen any radiation or chemotherapy, what are the mutation profiles? And this is what we see. Again, what the important finding here was that the major mutations that we see, histone mutation, P53, PPA1D, at postmortem, they actually exist up front, before any radiation, before any chemotherapy. This is the profile of all of the patients. Again, genomic aberrations as shown here in this circus plots. And this is the schema of how patients were treated. This is a patient 11, for example. The patient has PDGFRA amplification and received mebendazole. If the majority of the patients had H3F3A mutation, this is histone 3K27M mutation, they received panobinostat. Again, based on their mutation profiling, the patient received the following drugs. And this shows the feasibility phase of the trial where the patients were treated based on their primary mutations. This manuscript is being reviewed and we are hoping that it will be published soon. But if you look at the primary, so what we learned from this tumor is really interesting, and I think this is really one of the most important slides in this talk. This is a primary tumor, and it's showing the genomic aberrations of the primary tumor. Upfront biopsy that was taken from the patient. At the progression, some of the aberrations have changed. If you look at the primary cell line has the major mutations, but has new mutations. This tells us that some of the cell lines and mouse models that we test in our laboratory may not be representing the actual tumor. They may have additional mutations because we are doing clonal selection. But more importantly, look what happens at the postmortem. This is postmortem, but different areas of the brain of the same patient. Again, major mutations have stayed, but the tumor has really changed. This is left pons anterior, and this is interior medulla left. These are different tumors. Even if Sergey goes in with a CED and panobinostat and gets rid of the tumor within the pons, I think we're going to have some major issues of how to treat these other tumors that have metastasized, unless if we can catch this tumor earlier, perhaps in a combination of CED with immunotherapy. We take, as I mentioned, we also take these cells and we make mouse models of this. This is a mouse brain. This is a pons. The mouse shows exact histology of the human. It is positive for K27M mutation, K27M trimethylation, also positive for mutation. We also grow cell cultures. And we take the mouse and the cell cultures and we give them this exactly same treatment, same drug treatment that the patient was receiving, and we change the treatment to see if the outcome of the patient would be different. But then the question is that what would happen if you would actually put this drug directly into the mouse brain? If you would make an avatar model, a model of that patient, and then instead of giving the drug orally, you would put it directly into that. This is the work of one of our neurosurgeons. This mouse is a xenograft, has a tumor of human tumor. This is a subcutaneous pump. This is an osmotic pump that will release the drug over 2 months. It stays under the skin, is subcutaneous, and we study this to see what actually happens to this. Again, this is the end of the head of the catheter that is sitting within the pump. Here we injected dye, blue dye, to see what is the diffusion rate, because we needed to adjust the rate to get the right rate. So we are testing this with panobinostat, with other drugs that our patients are putting on, specifically because the next phase of PNOC trial will be a CED. We are going to have actually pumps in our DIPG patients that we're going to be testing. This is an example of, again, the diffusion. It stays within the pons. It's actually a beautiful diffusion of direct injection within the pons. And this is, again, injection area of our dye. But one of the major questions, and I always have this problem with our clinical colleagues, that they look at the genomic mutations and they select drugs to give to a patient. So they select a drug basket based on genomic mutations. But if you ask them, does this drug crosses the blood-brain barrier or not, they will say, we don't know. So we actually give a combination of drugs that could be toxic to our kids, but we don't know the penetration of that drug. What we've done here in our laboratory is that we've taken mouse, again, Sergey, these are our neurosurgeons who actually come to the facility and do this for us. They have put in a pump into the brain stem of the mouse. In the blue line here, we inject artificial CSF into the brain, and it washes out. So we feed the mouse with any drugs that we want to test, and CSF is washed out into this tube, and we can test it. We can look to see if it crossed the barrier, and if it did, how long did it take it to metabolize? We look at PK, PD, and also the crossing of the drug into the blood-brain barrier. Again, there's a lot of room for improvement and testing different drugs that we need to know if they cross the barrier or not. But surgery is invasive and is not an option, and it's 2008. I've talked about this before. Alex totally agrees with me. You guys sent the first man to space in 1961. 2016, we landed a shuttle on an asteroid, which is amazing. And in 2018, we have done really nothing for a tumor that is 2 centimeters below. Surgery is an option not for DIPGs, but really for most of the CNS tumors, we haven't done much. You didn't hear many success stories today. Specifically for brainstem tumors, we don't have any success story. In 2018, I think we should be doing, but again, a good restaurant has a good kitchen, a good hospital should have a good research. Liquid biopsy is what we have tried to establish, and your colleagues here are pioneering it in Russia. Amila and Alex are doing this. This is taking advantage of blood, urine, and CSF with the idea that you can actually take this liquid and look at the DNA that has been shed by the tumor cells. We isolate ctDNA circulating biomarkers, we amplify them, and we identify them. Liquid biopsy will hopefully bring up precision medicine. Again, prior to taking a biopsy of the patient, which is surgical, we should be able to take urine or plasma and be able to identify this. These are different mutations that we have been identifying so far. If you take a plasma of a patient that has this mutation in the brain, we can actually identify them. H3, F3A, H33, H31, ACVR, and PPM1D, and a few of the P53 mutations. This is how it looks. When you run a plasma or CSF of the patient, you can actually calculate the wild type and mutant, and you can calculate allelic frequency. You will know if the patient is homozygous or heterozygous for the disease. Again, examples, histone 3, ACVR, and PPM1D mutations that you can identify. This is another cool thing that we did. So again, when surgeons take a biopsy, it's really tiny. Many of you have seen it. It's a minute biopsy that is sent to the pathology, and the pathologists will scrape off that piece of tissue, and then the tefla, the gauze that the tissue has come in, they will throw away the gauze. We go and collect that gauze, we take it back to the laboratory, and we rinse it off, and we do the dPCR. This is a piece of trash for pathology that we took it to the lab, we washed it off, and the same day we could tell that this patient has histone 3 mutation from a piece of trash. So pathology knows now. Every time they get the gauze from their neurosurgery, they scrape off the tissue that they want, and they freeze this for us. We actually keep this in our freezers because you can go back to it and get a ton of genomic information from this. You can also do multiplexing. You can look at multiple mutations. Here we're looking at ACVR, wild type, mutant, histone 3 wild type mutant. Again, because these are very valuable tissue, 1 ml of CSF or plasma of the patient that you acquire, you can do multiplexing and see it. This is a case of a 16-year-old patient who wasn't surgery, had a progressive headache, CSF was collected, and we analyzed CSF, and you could tell that this patient is homozygous for histone 3 mutation. Again, prior to any, CSF was collected because the patient had hydrocephaly. And CSF is a very good medium actually for genomic information. You could tell the patient has this mutation prior to any surgical procedure was performed on this patient. This is a cohort of PNOC patients that I showed you, the DIPG patients, the 36 patients that were enrolled. You can see this is at the time of biopsy. This is the amount of the DNA, the mutant DNA that you see. It drops after radiation in majority of them and stays low throughout. Look at this patient. It drops after radiation, stays low during the treatment, spikes up between the two treatments, and then goes down again after the second treatment. So you can actually do clinical monitoring of the patient. You can't possibly take serial biopsies of the tumor, but you can do serial biopsies of the plasma. And if you compare this to the volumetric size of the tumor, this is as assessed by the MRI, this is as assessed by liquid biopsy. You can actually see that both are correlative. But we need to think big. What we have done at Children's National, we have teamed up with the CBTTC, Children's Brain Tumor Tissue Consortium. These are different countries in the world that we work with. The idea is that all of the genomic mutations should be shared with everyone. The same concept that Uber is the largest taxi company in the world but doesn't own a single car. We shouldn't be owning genomic information. We should be sharing them. And the idea of CBTCC is that we will share all of the samples across and all of the genomic DNA. Australia is our new site, and we have over 1 petabyte of data that is on the cloud and can be shared. Again, institutions who want to join CBTCC, we will be happy to share all of the genomic information with them on the cloud. They can download and they can access information. We have over 25,000 specimens. These are all pediatric brain tumors. 25,000 patients that have been banked at the CBTTC. And data is open. Again, this is Project Open DIPG that I'm leading in Washington, D.C. The idea is that we will have everything. For DIPG specifically, for high-grade gliomas, we have 838 DIPG genome. This is a whole genome, RNA sequencing, methylation, histology, MRI that will be available, again, being available. This is tumor types, mutations, histone 3 mutations, or H3K27, H3, H3, H3 wild type, or none of the histone mutation, histone wild type patients. Demographic distribution of the patients. And again, some of the primary whole genome analysis data of the patients that we have done, or 800 patients. We've connected laboratories across the different continents such that any investigator or any clinician can ask a question and can have access to MRI files or genomic information. I think this is the thing that Alex and I have agreed, that we need to quickly identify the biology. I started the talk saying that we don't really know much about the biology of the tumor. We think we do, but we really don't. We haven't connected the proteomics to genomic, for example. We need to make cancer treatment personalized, and we really need to use technology. Technology is, again, if you really have a good rice cooker, you make the best rice in your kitchen. We need to get technology to our laboratories, and Galina has done a great job with pushing forward with establishing a good laboratory here that can do both clinical and basic research work. And we are hoping to do this through collaboration with Dmitry Rogachev and also Bordenko at some point. We should be able to getting our powers and getting our resources together in order to expedite pediatric disease research. And thank you very much. Spasibo.